Well, it's my pleasure to introduce our speaker for today, Dr. Manish Shrivastava. Manish got his PhD degree at Carnegie Mellon University in 2007. And for more than a decade now, he's been a scientist at Pacific Northwest National Laboratory, DOE lab in Washington State, where he's now a senior staff scientist and a project manager. And Manish is a recipient of the DOE Early Career Award, and through that he's been exploring aerosols and their impacts on the Earth system, and including the work that he'll be talking about today, which comes from DOE and other investigators' work down in the Amazon. And as soon as we have this Zoom system, so this is our ESS is getting into the hybrid thing here. So we have, typically we've had about as many online as here in the room. So we'll try to serve that part of the audience too, but not that you guys aren't important. Okay, thanks, Alex. And so take it away, Manish.
Thanks, Alex, for this nice introduction, and thank you all for coming to the seminar. It's always a privilege coming to you here and meeting such an esteemed group of colleagues and students. So the work which I'm going to present today is about an integrated model measurement study, which we did over a couple of years now. And the goal of this study was understanding SOA formation. And in this case, I am focusing on isoprene epoxydiol SOA. So over the Amazon region, isoprene, as we know, is one of the most abundant VOCs emitted, and it has a lot of role to play in atmospheric chemistry and SOA formation. So I want to, this was just published recently, and I want to acknowledge all my co-authors, including Alex here. I also want to acknowledge the US Department of Energy's Office of Science, Biological and Environmental Research Early Career Award, and ASR for funding.
It's still not sharing. I'm so sorry. I'm so sorry.
Okay. So this is just a very general slide about, why do we want to study secondary organic aerosols and what are they? So mostly we know, like the way we distinguish, we have primary organic aerosols, which are directly emitted from various sources. It could be vehicles, it could be power plants, so these are directly emitted organic species in the particle phase. And the way SOA or secondary organic aerosol is different is because it starts its lifetime in the gas phase. So you have various kind of volatile organic compounds, isoprene, terpenes, and also many other VOCs emitted from anthropogenic sources. And these react in the atmosphere and make thousands of oxidation products. Some of them can condense directly on particles, the others undergo multi-phase chemistry. So the reaction between gas phase VOCs and particle phase components. For example, in this case, I am talking about IPOX SOA, which is one example of multi-phase chemistry. It needs chemistry both in the gas phase and particle phase to form this species. When we think about other species like sulfate and black carbon, like most of the climate modelers, think about them, they are mostly one species. Like you could think of sulfate as one species, black carbon as one species. But SOA, even if we refer to it as one species, it's not one species because each particle of SOA is made of thousands of organic compounds, and these the composition of these particles can change dynamically in the atmosphere. And the reason SOA is really important is, you may have very small clusters of nuclei which are found, maybe it's sulfate or some highly oxidized organic molecules. But to facilitate the growth of these clusters to sizes which are relevant for clouds and cloud condensation nuclei sizes, organics and SOA plays a central role. And so in this way, they affect radiation and clouds, and they are really important to the atmosphere. Okay, so this is just another chart from the IPCC radiative forcing. And so on the top, you have all the greenhouse gases, and then, they are warming and then you have, in the central portion, you have things like black carbon and organic aerosols. But the main point here is that the organic carbon, which was included in the IPCC radiative forcing bar chart, it's mostly primary. And SOA was not even included in the radiative forcing estimates because the previous IPCC report acknowledged that there is tremendous complexity and uncertainty in the processes involved in SOA formation. So even though the radiative forcing bar chart talks about organic carbon, it's primary, it's not the secondary component of organic aerosol. And so I would expect, like, given that the SOA is ubiquitous along in the globe, you would expect that the direct radiative forcing of SOA is at least similar or greater than the primary organic carbon. But even more importantly, the role of SOA in cloud forcing is more uncertain because it can interact with CCN and clouds. There is also aqueous chemistry and cloud chemistry going on. And so the the SOA cloud interactions are not well understood. That's one of the things about this, which I wanted to mention. And this is just a figure from our reviews of geophysics paper that came out in 2017, where we are showing one of the main points of this paper is that we cannot think of biogenic sources separate from anthropogenic sources, right? You have biogenic VOCs doing their own thing, making SOA, and anthropogenic SOA being formed in its own respect. But in actuality, in the atmosphere, these interact together. And the interactions between anthropogenic and biogenic VOCs is what, there are a variety of pathways. It could be through sulfate, NOx, but even interactions among organic molecules coming from anthropogenic and biogenic sources. And these interactions are important because they affect the phase state of SOA, whether it's liquid, solid, or semi-solid in the atmosphere. And that really affects how much more SOA can form. So because that affects the viscosity of these particles and also their volatility and lifetimes. So if you have a solid particle, for example, of SOA, the diffusion timescales within that would be very large. And so it would be harder to make more SOA on top of it in the conventional volatility frameworks. Whereas if you have a liquid-like particle, it can easily absorb more organic mass and you make more SOA. And so that feeds back into new particle formation, growth and evaporation kinetics, but also the multi-phase chemistry, which is chemistry that happens both in gas and particle phases. And the reason it is important is, again, it affects CCN number, the optical properties, the climate relevant properties of these particles, which interact with clouds and radiation, and then feed back to biogenic VOC emissions. So you could see that the cycle is complex and it's mainly the anthropogenic biogenic emissions which are not well understood. And also they are not included in many of the regional and global models. And this could bias the radiative forcing of aerosols. So given this motivation in mind right now, I'm going to focus on one component of SOA, which has received a lot of attention in the past decade. And that's about isoprene epoxidiols. About a couple of decades ago, it was thought that isoprene does not make much SOA. The previous chamber studies in the 80s were not making much SOA from isoprene itself. But then a decade ago, there were these findings about isoprene epoxydiol SOA, which is made by multi-phase chemistry. So you need gas phase chemistry like isoprene oxidizes. After 2 generations, you make ipox in the gas phase, but then you need certain ingredients in the particle phase to make ipox SOA, because once the ipox gas is taken up in the particle phase, there is an acid catalyzed ring opening and then followed by nucleophilic addition of sulfate and water. And that's what makes the SOA in the particle phase. So you have like both gas phase and particle phase chemistry being equally contributing to ipox SOA. And there are, if you look here, like this is from the WHO et al. 2015 paper, there were several places. The pie charts are in the green. That is the component of ipox SOA in the pie charts, and then the shaded regions are just geochem predictions of ipox gas. So the key is that now it is well recognized that it is forming ipox SOA in many places, including over Amazon, over Southeast USA. It is shown to be really important, the ipox SOA formation chemistry. But still, the point I want to make is that many models are still not treating the ipox SOA processes correctly. They are missing many key processes, including the viscosity of organic aerosols, which is really important. And so this is just a figure from our paper where it's showing that you have a core of inorganic aerosols at the center, which has sulfate, ammonium, water. It's acidic. It could be less acidic or more acidic. But then the reactive uptake of gas phase ipox within this core depends on its diffusion, the mass accommodation coefficient on the surface. But then also, as the ipox SOA starts forming, even if there is no other SOA around, as ipox SOA starts forming around this core, it itself is highly viscous. So then it starts self-limiting further uptake of ipox SOA within the core. And then if you are in the real environment, you have many other species of SOA. You have monoterpene SOA, sesquiterpene SOA, and all those, if they are forming a coating, then that will limit the uptake of ipox. And then also another key ingredient is that you need water in the particle phase in the core to make, for the aqueous chemistry to happen. So there are several processes when as modelers we try to model the ipox SOA formation, need to think about several processes and their uncertainties. Diffusion limitations in the organic shell is one of them. There is chemical kinetics in the inorganic core that also needs to be well constrained by measurements. But then getting the total organic aerosol concentrations right. What is the thickness of the coating? What is the viscosity of the coating material? That is really important. Then obviously isoprene emissions are important, because if you have, depending upon how well you are modeling the isoprene or getting the isoprene emissions right, that could change the availability of gas phase ipox. And then ultimately, in addition to aqueous phase chemistry, you have cloud phase chemistry. So you could think of the ipox SOA forming, becoming a CCN, going into the cloud droplets, but then also there is an uptake of ipox in the clouds. And even though the reaction kinetics are much slower in the clouds, the reaction volumes are large, because you have large water content in the clouds. So you will add some ipox in the clouds, and then there is when the cloud droplet evaporates, you have the aerosol coming out, and there is a cycling between cloud phase and particle phase here. So these are complex processes which we need to think about when we think about modeling ipox SOA formation. So this is just to introduce the concept of viscosity here, including Manabu was a co-author on one of these papers. But if you are going from water all the way to olive oil, honey, all the way to marbles, you are increasing the viscosity of that fluid. So since SOA can exist in a range of spectrum, it could be liquid near the surface, it could be solid in the upper troposphere. So it goes through a range of these phase states. And on the graph which I'm showing, you have diffusion coefficient on the x-axis, you have particle diameter on the y-axis. And if you think in the semi-solid regime, if you have a 1 nanometer particle, maybe the diffusion timescales would be a minute or less. But if you have like 100 or 200 nanometer particle, which has grown then the diffusion timescales can go up to a day or so. So in addition to viscosity, the size of the particle matters as it is growing. So that's one of the reason I'm showing this. Then coming back to how we actually model IPOX SOA. So one way to model it is using core shell morphology. So in the core you have inorganics, and then in the shell you have the organics coating it. And so the way you get the reactive uptake coefficient of IPOX gas by solving the spherical reaction and diffusion equations here. And then on top of that, you do a flux balance between across different interfaces, across the organic air interface and the organic aqueous interface. And when you do this flux balance and solve the reaction and diffusion kinetic equations, then you end up with a overall reactive uptake coefficient, which has several terms on it, including gas phase diffusion, mass accommodation coefficient, but then also it's the reaction and diffusion occurring within the inorganic core, which has water inside it. So that is the aqueous term. And then you also have the reaction and diffusion happening in the coating, the organic coating itself. So one of the things which is really important is we have to go back to actual measurements and try to see how well do we understand the kinetics of IPOX SOA formation. So this was another paper which came out from a group recently where we had single particle measurements in a Teflon chamber at EMCEL, where we were, so this was led by Ala Zelenyuk and co-authors like Jason Surratt and Riva. So what they were doing is you had ammonium bisulfate seeds, which are put into a 1 meter cube Teflon chamber. And then on top of that, you inject IPOX gas. And then you are monitoring the changes in the size distribution of particles as you are forming IPOX SOA on these acidic seeds of ammonium bisulfate. It was measured both by a DMA, but also the mini splat was measuring single particle composition, density, and also volatility change. And this was done both at dry conditions and wet conditions. So I'm showing the dry condition experiment here. This is 5% RH, but since it is ammonium bisulfate, it does not effloresce. So it always has some water content, even at 5% relative humidity, and it is highly acidic. So the pH is negative 3.5 or so. But then once you inject the IPOX gas and it starts forming IPOX SOA, one component of that IPOX SOA is organosulfates. So once you make organosulfates, you are removing the inorganic sulfate from the particles and converting them to organosulfates. And what it does is it decreases the acidity a lot. So if you look at the acidity plot here on the red line, once you are forming the organosulfates, you are converting the inorganic sulfate to organosulfate, the acidity is dropping by more than 2 pH units. Whereas many of the models, they do not treat the change in the acidity or the conversion of inorganic sulfate to organosulfate. So they will keep it artificially at very high acidity. And so when we compare box model simulations where the blue curve, so if you look at the size distribution plots, this is after allowing the IPOX SOA to grow and form after 2 hours. So the black is measurements, the final measured size distribution there, and the blue line there is the models which do not account for organosulfate formation. So they keep the acidity high. So you could artificially make too much IPOX SOA. As you can see, the size distribution is shifted too much to the right. Whereas if you account for the conversion of sulfate to organosulfate, you see much better agreement, either by the red size distribution or the green lines there. And in addition to the constraints by size distribution, we can also compare the changes in the density of aerosol as you are forming IPOX SOA. You can look at volatility changes. So what the measurements were showing, the single particle measurements, that about 90% of IPOX SOA was non-volatile. So that gives another constraint, because when you make IPOX SOA, you make 2 components. One of them is tetral component, which is due to water as nucleophile, and the other is organosulfate, which is of very low volatility. And tetrals are semi-volatile, so some of them will partition back to the gas phase. But in the particles, some of them can also make oligomers, which is of low volatility. So the point here is that when you have multiple measurements of size, composition, density, volatility, and then also the mass spectral measurements, you can really use that to constrain what you know about the formation of ipox SOA processes and the chemical kinetics. So that was just that was validation of some of the ipox SOA mechanisms with laboratory studies. So this is now I'm going back to trying to model the Amazon field campaign. During the Go Amazon 2014 field campaign, there were measurements by the DOE G1 aircraft, but also the German Hello aircraft was flying. And the German aircraft was flying all the way from near the surface to about 12 to 14 kilometer altitude. And so this huge altitude range was a very nice constraint to understand the processes in the models. The way I'm modeling it, WorkCam 4.2, again, like you can see the modeling domain over the Amazon here. And the G1 aircraft had the PTRMS instrument on board. So that gave a nice constraint about isoprene emissions. So initially when I started modeling it, WorkCam model was too high by a factor of 3 with respect to isoprene emissions. So we had to bring it down because I think it's really important to constrain the emissions also in addition to the mechanisms. And we have, over the last few years, we have made several updates to SOA chemistry, new particle formation mechanisms, which we have published in different papers. And then also like building up with the box model evaluations, which I showed in the slide before this, because we need to evaluate our processes with actual controlled laboratory measurements to be certain that we are going in the right direction here. So the graph below that, that's just a plot of isoprene emission fluxes over the Amazon. And this is the picture of the German Hello aircraft is shown. But the key point here is that the vertical profile, this shows the organic aerosol profile measured by the aerosol mass spectrometer over the Amazon. This is averaged across several flights during September of 2014. So the key feature here is the observed organic aerosols are either peaking near the surface, like less than 2 kilometers or so, or there is another peak you could see above 10 kilometers in the upper troposphere. And in the middle troposphere, you see them dropping significantly. And so the main thing is we want to understand what causes this bimodal peak. You have, obviously near the surface, you have emissions and you have more chemistry going on. But then even in the upper troposphere, then what is causing the peak in organic aerosols and ipox SOA? Because in the upper troposphere, the temperatures are really low. It's 250 Kelvin, minus 50 degrees Celsius or so. So that means that at such low temperatures, whatever organic aerosol is existing, whether it's ipox SOA or monoterpene SOA, it is mostly in the solid phase. So I'm going to talk to you about that as well. So in WorkCam, we implemented a parameterization. Again, it's coming from Manabu's work, where we can predict the viscosity and glass transition temperature of organic aerosols. So we are doing it online now because in WorkCam, we have several mechanisms which are making different kinds of SOA. There is biomass burning organic aerosols, there is isoprene SOA, there is monoterpene SOA, HOMS, new particle formation, all these mechanisms we have developed and implemented in WorkCam. But then what is the overall viscosity of this organic mixture? And how does that vary as a function of altitudes, as a function of temperature and relative humidity? So we found that, near the surface over the Amazon, humidity is high, 70 to 80%, temperatures are warm. So the model is predicting that SOA is liquid-like. But if you look in the upper troposphere above like 7 or 8 kilometers altitudes, then we are predicting SOA is mostly solid because of low relative humidity and very low temperatures in the upper troposphere. And another finding from this work, which also came out recently, is that if you take the log of viscosity and the logarithm of water, which is associated with organic aerosol, you get a very strong linear dependence. And this is over the entire atmospheric column over the Amazon. So in field studies, if you don't have direct measurements of viscosity and you can estimate the water associated with organic aerosol based on its hygroscopicity and other things, then you can, we think that you could figure out the viscosity to fairly good degree. Okay, so now going back to the WRF-Chem simulations. So I started this work about a couple of years ago. And here I'm showing on the leftmost, I'm showing 3 altitude ranges, like because in the middle troposphere, the aerosol concentrations were very low. So we are only focusing on near surface and upper troposphere. And so if you look at 0 to 2 kilometers, the y-axis is IPOX SOA mass concentration averaged across a given flight. And so between observed and default in blue, you get like factor of 2 or 3 difference. But if the main thing was, if you look in the upper troposphere, like 10 to 12 kilometer or 12 to 14 kilometer, there the model really struggled. So we could not make, I mean, the IPOX SOA, which the model predicted, was 10 times lower than measurements. And that was a huge discrepancy. And there was very real reasons for that. Because if you look at the viscosity plot, like near surface, it is all liquid light. But at 12 kilometer altitude, the viscosity, the organic aerosol is solid. So if the aerosol is solid, there is no, I mean, you have very strong diffusion limitations. So IPOX will not be able to go through the shell of solid organic aerosol and then react with whatever is in the pore. That was one thing. And the other thing also is that because the relative humidity is low, we have like about 30% RH. So most of these salts, even inorganic salts, they don't deliquesce at that low relative humidity conditions. So we were getting, like, we were running the mosaic inorganic model here in WRF-Chem, and that was predicting 0 water in the particle phase. So if there is no aerosol liquid water, there is no way there can be aqueous chemistry. So that was a major conundrum. When I was first started modeling this, I was like, okay, there is no way I'm getting IPOX SOA in the upper troposphere. There are 2 main issues to it: this solid shell of organic aerosol and then also no liquid water. So then why is the aircraft actually, why is it seeing IPOX SOA in the upper troposphere? And that was a major conundrum for some time. So I'm going to like tell you a story about how, you know, this thing evolved. So I was at the AMS meeting at some point, and then somebody was presenting from some of measurements from Alex and Scott Martin's group, where there were this possibility of direct measurements of methyl tetrals. So now if you condense methyl tetrals, the AMS will measure them as IPOX SOA because they are one of the major components of IPOX SOA. So over the Amazon, like with the UAVs, you know, this paper is showing that the two methyl tetrals are varying by more than 480% over hundreds of meters, and the heterogeneity, even if the isoprene emissions are heterogeneous and their atmospheric oxidation happens, that does not explain these differences in 2 methyl tetrals because they have a longer atmospheric lifetime here. So the direct emissions of 2 methyl tetral gases, which are modulated both by the type of forest and environmental stressors, was proposed to be the best explanation. This was a 2021 paper. So I was like, okay, this sounds like, you know, a great mechanism, and maybe it can help because there is no other way I'm getting agreement. And so it was like connecting the dots between different measurements and process level models. So when you include the methyl tetral, so there is uncertainty in methyl tetral emissions, but the paper came out with some limits. So in my work, I used the lower bound of methyl tetral emissions to include in WRF-Chem. And so if you add the methyl tetral emissions in the WRF-Chem model, you still see, like compared to IPOX gas, near the surface methyl tetrals are 10 times lower in concentrations. And in the even in the upper troposphere, methyl tetrals are 4 times lower. So it's not like you are bombarding the model with too much methyl tetral gases. You know, it still sounds pretty reasonable because IPOX gas is much more abundant throughout the atmosphere. But the major thing to keep in mind is IPOX is highly volatile. It has 3 orders of magnitude higher volatility than 2 methyl tetrals. Because 2 methyl tetrals are semi-volatile, they can easily partition to the gas phase, both near the surface and the upper troposphere. So in the model, when you include the gas particle partitioning of methyl tetrals, you see that near the surface at 0 to 2 kilometers, the average fraction of methyl tetrals partition to the particle phase is 60%. So 40% is in the gas phase. 60% of them are in the particle phase because temperatures are much warmer. But over the high altitudes, what happens is that in Amazon, the deep convective events, they are transferring these gases all the way to the upper troposphere. And where cloud dethronement occurs at 10 to 12 kilometers, it's very cold, like negative 50 degrees Celsius. And so at that cold temperatures, whatever methyl tetrol gas was there, it will all condense to particle phase. So getting like 99.9% in the particle phase in the upper troposphere. So they are very efficient in particle production just by direct condensation at those low temperatures. And the reality is that all the models which we know of, which I know of, none of them include these direct emission sources of methyl tetrals. So it's a very important source. If the forest is emitting them by direct biological processes, in this study, we found that their concentrations in the upper troposphere, they are more than 90% of IPOX SOA, but even near the surface, their source is larger than atmospheric chemistry alone. So far, like it is believed that atmospheric chemistry is the major source of IPOX SOA. But when you include this and you constrain it by upper tropospheric measurements, right? Because if it was not for the upper troposphere, you could think that, oh, you don't know the kinetics well near the surface. You don't know the diffusion coefficient well near the surface. There are a lot of uncertainties if you're just looking at the surface. But when you really look in the upper troposphere, where there is major differences between the model and measurements, and you know pretty well there that diffusion limitations are real, the particles are solid, relative humidity is low. And so the only way you could explain it is with these direct emissions. So going back, in WRF-Chem, we are modeling the cloud. So this is a 10-kilometer grid spacing. It still doesn't get all the clouds, but we had to run convective parameterizations. So WRF-Chem simulates cloud top heights either at 1.5 to 2 kilometer or deep convection that extends all the way to greater than 12 kilometer altitudes. So when you have deep convection events, the cloud where they entrain, that's at the upper troposphere between 10 to 14 kilometers. Another interesting finding from this work, which I'm not presenting here, is you have a lot of new particle formation due to monoterpenes in the upper troposphere. You have like 50,000 particles per cc. But if you turn deep convection off, you will not be able to make those particles. So deep convection is actually coupling the surface to the upper troposphere. There is much less clouds in the middle troposphere, and then consistently, when you look at AMS organic aerosol measurements, you see two peaks, one near the surface and one at the upper, at 10 to 12 kilometer altitudes. So this is some validation because the Halo aircraft, the German aircraft, was also measuring things like temperature and relative humidity. So it's really important to get these things right in the models also. So if you look at some other global models, sometimes they have like 70% relative humidity in the upper troposphere. Obviously, that is not right? Because at least in this region, the bars here are model and the dots which I'm showing are observations. So ambient temperature, both, like for example, at 12 to 14 kilometers, you get 200 Kelvin, and the model agrees with measurements. And then also for relative humidity, it's less than 40%, both in model and measurements. So it's not going to 70% because of course, if you have 70 to 80% relative humidity, then you can have liquid water in the particle phase. So it's important to, when you are modeling it, it's important to also evaluate the temperature and relative humidity profiles with aircraft measurements. And here WRF-Chem was doing pretty well. And then also the ratio of blast transition temperature to ambient temperature, if it is less than 0.8, it is liquid-like organic aerosol. If it's 1 or greater than 1, it is solid. So the bottommost panel, the C panel, you are seeing that near the surface 0 to 2 kilometers, you get liquid organic aerosol. In the upper troposphere, you get solid. So to further understand what is, so in this case, the bars I'm showing here, the gray bars, those are actual observations from the Halo aircraft at 3 different altitudes. And then I'm showing 2 sensitivity studies. The green bar has atmospheric chemistry in there for IPOX SOA, but then it also has a source of direct emissions of methyl tetrals. And you see, especially if you focus on the upper troposphere, like 10 to 12 or 12 to 14 kilometer, it comes pretty close to the observations. And then there was another sensitivity which I did where you can still have deep convection on in the model, but you turn off the convective transport of tracers due to deep convection, and that is the red. So if you turn off the deep convective transport of tracers, then you will not get the IPOX SOA which you are seeing in the upper troposphere. And that is another proof that you need, in addition to atmospheric chemistry, direct emissions, you also need this deep convection events, which actually are really important to explaining what is happening in the upper troposphere. And then also, like, if you look at the pie charts, we see that near the surface, the organic aerosol composition is more diverse than the upper troposphere. So near the surface, you have biomass burning, you have, in addition to IPOX SOA, you have other isoprene SOA, you have monoterpene SOA. But if you look in the upper troposphere, about 70% is monoterpene SOA, and you still have 10% IPOX SOA. So it's less diverse in the upper troposphere, much more diverse near the surface. So that's why, because of the diversity of organic aerosol and because of other uncertainties, what really jumped out was looking at the upper troposphere gave critical clues about how well you can constrain or understand the processes of IPOX SOA formation. So this is one example. This is from a previous study where we are, Ben Zhao was leading it, and we were showing that in WRF-Chem we implemented the two-dimensional volatility basis set for monoterpenes. So that accounts for HOM formation and their temperature-dependent auto-oxidation and the dependence on NOx. So if you look at the total number concentrations of particles, they peak strongly in the upper troposphere. And with the right nucleation mechanisms and HOM formation, you can reproduce that high particle number concentration. So I'm showing this figure because in the upper troposphere, the model was predicting that about 70% was monoterpene. So that is not realistic based on, that is realistic based on measurements of total particle number concentrations, which we have seen before. So this is just a recap of several model sensitivity studies, because we were thinking about a new mechanism which the atmospheric chemistry community has not considered so far, like with the direct emissions of methyl tetrals. So we really had to make it rock solid. So I have done several different sensitivity studies in WRF-Chem. So one of them is, what if we treat the particles as liquid-like in the upper troposphere so that they still can retain particle water, and then the organic shell is still liquid-like. So can that explain IPOX SOA, right? And then there are other things where you emit the tetrals, where you turn off the deep convective transport, and then the another sensitivity was, what if I just increase the reactive uptake coefficient or the chemical kinetics in the inorganic core? So how do each of these sensitivity studies affect IPOX SOA concentrations? So this is just, just showing a recap of the sensitivity studies. You have the observed as the gray bar, and the blue is the default model. As you can see, if you focus on the upper troposphere, the model is at least an order of magnitude lower than observations. And so now what you do is, if you treat the organic aerosol as liquid-like in the upper troposphere, artificially by setting its diffusion coefficient to be equal to water, and then you also allow the particles to retain liquid water in the particle phase. That is the yellow line. So then you can explain measurements in the upper troposphere, right? So this is showing that these two mechanisms, where the primary diffusion limitations and existence of water in the particles, that was really the contributing factor to create model measurement differences. And this one, the magenta line now, is you still treat the particle as diffusion limited. There is not much particle water, but you increase the reactive uptake coefficient in the aqueous inorganic core. So you can increase it near the surface, right, as you can see in the magenta line, but in the upper troposphere, you again have at least an order of magnitude difference. So just by increasing the reactive uptake coefficient, you increase what forms near the surface, but you cannot increase in the upper troposphere because of strong diffusion limitations and also because of absence of particle liquid water. And the green line is once we emit the methyl tetrals directly from the forest, like we are emitting isoprene. And so then the green line, now the green line has all the realistic processes. It has diffusion limitations in organic shell. It also has almost zero liquid water content, like you would expect at low relative humidity. But the direct emissions of methyl tetrals, as I explained, when they are transported by deep convection, they can condense directly and make these particles. So that comes pretty close to measurements. And then the red line is when you still have emissions of methyl tetrals, but then you just turn the transport of tracers by because the deep convection is transporting all the tracers to the upper troposphere. So in the model, you could still have deep convection on, but you could turn off the convective transport of tracers. And once you do that in the red line, you see that again in the upper troposphere, you cannot explain IPOX SOA. So that is really saying that all these processes, diffusion limitations in the organic shell, realistically treating relative humidity and particle liquid water, constraining the IPOX reactive uptake as much as you can, and then deep convection along with methyl tetral emissions, all these processes you needed to think about, and isoprene emissions, of course, to try to get the right answers and for the right reasons in the models. Very few models, do such, because this took us more than a couple of years working through all these processes. And this is, I'm just showing you the surface distributions now of IPOX SOA. So the leftmost panels are the default model where you don't add any emissions of methyl tetrals. And you could see like at 0 to, like if you look at the bottom panel, the leftmost bottom panel, you see at 0 to 0.5 near the surface, you see most IPOX is following the plumes. You have the plumes from Manaus going in. So because that's where in the plumes you have more sulfate and liquid water. So you will make more IPOX SOA over the plumes. But the reality was when the aircraft is measuring IPOX SOA, it's flying all over near the surface and upper troposphere. And everywhere it flies, it sees almost uniform concentrations of IPOX SOA or higher. So that is another evidence that once you put in the methyl tetral emissions in the middle panel, you see almost ubiquitous sources of IPOX SOA, both near the surface and upper troposphere. Especially for the upper troposphere, the contrast is huge, right? At 13-kilometer altitude, the default model has, in the top panel, it has almost no IPOX SOA. Whereas when you have emissions of methyl tetrals, you make more IPOX SOA. And when you turn off convective transport, it again goes down to 0. So this is just another figure here, which is showing you that you have these direct emissions of methyl tetrals being carried up by cloud updrafts. And in the upper troposphere, there is no diffusion, no cloud chemistry, no liquid water in the particle phase, but then the direct condensation of methyl tetrals can explain, it can uniquely explain aircraft measurements because we have looked through it by conducting many sensitivity studies in the model. This is just an ongoing work which I'm doing. So as I mentioned, we had to increase the reactive uptake coefficient of IPOX SOA. But in reality, what we are finding is that at high RH, at 50% RH, even the reactive uptake coefficient should be 10 times lower, not 10 times higher. So this is a work in preparation, again, trying to explain single particle measurements at MCell, because at high RH, we see that the base case model is greatly over predicting IPOX SOA. So that is again arguing that you don't need to, the measurements are saying you don't have to go higher. In fact, you have to go lower. So you will see even more gaps between model and measurements if you don't include these direct sources of methyl tetrals. So this is my final slide. Just want to say, like, what I learned from this is that comparing the measurements and model predictions in several levels, like both laboratories and field experiments, is critical. They provide critical insights. And in this case, the upper troposphere, even though it was a big conundrum for me for at least a year or more, that really had critical clues about what is happening about IPOX SOA processes, which are not included in models. So at high altitudes, low relative humidity and temperature, lack of particle and cloud liquid water, and if the coating is solid, there is no way IPOX SOA can form in situ. So the direct emissions of methyl tetral gases from plants, which is related to environmental stressors, plant stressors, and their convective transport, and condensation was critical. So this biological source of 2-methyl tetrals, it's not well understood. So more measurements are needed to understand it, to measure it. But connecting in this case, like connecting the dots between different kind of measurements, right? Like when I could not explain it in the upper troposphere, trying to connect the dots with other measurements, which were suggesting the possibility of methyl tetral emissions in gases, and then thinking about how that changes IPOX SOA formation. That showed the critical role of land atmosphere cloud interaction processes, which are not included in models. And this has huge implications because methyl tetrals are just one semi-volatile gas component. There are many other semi-volatile gases. And so when we are rapidly deforesting places like the Amazon, we are perturbing the natural processes of fine particle formation. And these modulate both global warming effects, clouds, and precipitation. So if we don't even have these processes in the models, we cannot predict their impacts. And so this is why the connection between atmospheric chemistry, convective transport, and the processes which we can constrain and measure is really important. Thank you.
Questions for Manish?
Really nice seminar, really nice work. So a question, the way I understand it, you're using a core shell model where you've got the inorganic aqueous core and the organic plate on the outside. Would it make any difference to your model if instead of having a core shell model, you had phase separation where you had organic here and the liquid here? Sort of a test result.
Yeah, that is a very nice question. It will matter. But I think when we think about the upper troposphere, when the temperatures are that low, right, and if basically, and the relative humidity is low, like less than 30% or so, then in that case, not much water is there in the particle phase. And also, if it is not a complete shell, right, like maybe I think like if it's not completely covered by SOA, right, there are pockets where it is not. And then there is liquid-liquid phase separation. Yeah, that gets more complicated near the surface, I would say. But in the upper troposphere, again, like if even the clouds are ice clouds, right? So in that case, I think we have more confidence that these processes of diffusion limitations and absence of liquid water in particles is really governing. So thank you for your question. Yeah, Sajid.
Yeah, very nice. So why do you think this is a biological source of tetrols? Why cannot it be just, you have horizontal air transport, isoprene oxidizing products sticking temporarily to trees, temperature increasing, they get off, they just sit. They still form in the atmosphere, they just sit on the tree surface temporarily.
Yeah, so
Manish, can you repeat the question to the online audience?
yeah. So Sajay's question is that why is it a biological source? Why cannot just be isoprene oxidation creating these products, right? In the atmosphere?
Yeah, so but the products, let's say first generation products, semi-volatile, they can hold on to the surfaces of the trees a little bit. Yeah,
that's a good, I think. So main thing about methyl tetrals is that it has to be, the particle phase is somehow involved because it has to have water and acidity and other things. It's a acid catalyzed ring opening mechanism which forms. So right now we don't know of any direct gas phase chemistry mechanism which makes methyl tetrals. That is one thing. So you need a heterogeneous medium, whether it is leaves or it could be soils. The another possibility which we proposed in the paper is that if there is gas phase IPOX which is deposited on the surface of soils or leaves, and if there is a medium there which has sulfate and inorganics and acidity, that can release methyl tetrals. So yeah, that is a possibility, and that has to be explored more through measurements. But direct gas phase chemistry making 2-methyl tetrals, we don't know about that mechanism.
Nice work. I may have missed this Oktavini paper of this IFSSOA pH change. So you said that upon overall sulfate formation, pH will increase.
Yeah,
Why
because
is that?
we start with the seed of ammonium bisulfate.
Oh,
So it's a pure seed which is highly acidic at 5% relative humidity. So with the mosaic model or any other inorganic model, you get highly acidic seeds like negative 3.5 or something. And then once you start making IPOX SOA, the sulfate, the inorganic sulfate, that acts as a nucleophile, so that makes organosulfates. And so when you are removing the inorganic sulfate,
so you want bisulfate?
yeah, inorganic so you are removing from the bisulfate, yes, but then you are decreasing the acidity, right? So you are making more than whatever ammonium is there. I think it will increase the sulfate. It will go to ammonium sulfate over time. So that process, if models don't include that process, then they will maintain very high acidity and make too much IPOX SOA.
Is that measured by experiments?
The acidity itself was not measured by acidity is a calculation, but I think there were other things which were measured, like the size distribution change, the mass spectra measurements from SPLAT, and then also the changes in density and volatility of particles. I think Michael has a question.
Manish, on your earlier plots, you pointed out how important isoprene was for your aerosols, but it seems as though even in the middle of the tropical rainforest, isoprene is only 30% of the mass. What's the rest of it?
So you mean of the VOC mass, or?
Well, I don't know. You had your little breakdown of the pie charts.
Oh, that was just
only 30%. So more than 2/3 is coming from what? Is it inorganic? Is it just other VOCs?
So this is just the breakdown of organic aerosols, right, in the pie chart. So yes, so near the surface you have 20% IPOX SOA. In the upper troposphere you have 10% from the model, but there are other isoprene SOA which is not going through the IPOX pathway, right? So that's just gas phase chemistry. Isoprene reacting in the gas phase with OH, ozone, and nitrate radicals and forming SOA. But that does not need the aqueous phase chemistry pathway. And then you have monoterpenes and sesquiterpenes. That is lumped into the terpene SOA category. And near the surface, you also have biomass burning PO, primary and secondary organic aerosol. But in the upper troposphere at 12 to 14 kilometers, we see much less biomass burning contribution.
Okay, don't see anything online. Thank you, everyone. Let's thank Manish one more time.